Cells use information about their internal state and cues from the environment around them to decide whether to proceed with cell division. A checkpoint is a stage in the cell cycle when the cell examines internal and external cues and decides whether or not to move forward with cell division. The G1 checkpoint is the main decision point for a cell. Once the cell passes this checkpoint, it is committed to division. The cell checks to be sure that conditions are right for division. If a cell does not get the go-ahead at G1, it may leave the cell cycle and enter a resting period called G0 phase. The G2 checkpoint checks DNA integrity and DNA replication. If errors are found, the cell will pause and wait for repairs. If the damage is unfixable, the cell may undergo apoptosis, which is programmed cell death. The M checkpoint determines whether the sister chromatids are correctly attached to the microtubule spindle before the cell enters anaphase. Internal and external cues act by changing the activity of core cell cycle regulations inside the cell. Cell cycle regulators make sure that cell cycle events take place in the right order and that one phase triggers the next phase. Cyclins are the most important cell regulators. They must activate or inactivate many target proteins in the cell. A typical cyclin is present at low levels for most of the cycle, but increases strongly when it is needed. Cyclin-dependent kinases are paired or partnered with cyclins in the two drive parts of the cell cycle. Alone CDK is inactive, but the binding of a cyclin activates it. CDKs are enzymes that phosphorylate specific target proteins. Mitosis promoting factor stimulates the mitotic phase of the cell cycle. Once these complexes receive an additional signal, they become active and set the events of M phase into motion. Platelet-derived growth factors, PDGF, are known to signal cells by binding to a cell surface receptor that is a receptor to tyrosine kinase. PDGF stimulates fibroblast division not only in artificial conditions but in animal bodies as well. Cyclin is degraded results in degraded cyclin. Cancer develops from uncontrolled cell division. The development and progression of cancer is linked to a series of changes in the activity of the cell cycle regulators, which is due to mutations in the genes that encode cell cycle regulator proteins. Cancer cells make their own growth factors and ignore signals that should cause them to stop dividing. Cancer cells can divide many times because they express an enzyme called telomerase, which reverses the wearing down of chromosome ends that normally happens during each cell division. Cancer cells fail to undergo programmed cell death, apoptosis, under conditions when normal cells would. Most cancers arise as cells acquire a series of mutations that cause them to divide more quickly, escape internal and external controls on division, and avoid apoptosis. Normal cells undergo apoptosis if there is unfixable DNA damage. Whereas cancer cells continue dividing. There are two types of cell cycle regulators that undergo mutations that may promote development of cancer. Positive regulators may be overactivated, while negative regulators may be inactivated. Positive cell regulators may be overactive, such as growth factor receptors send signals even when growth factors are not there. Negative cell regulators may be less active or even non-functional, such as proteins that halt cell cycle progression in response to DNA damage may no longer sense damage or trigger a response. When there is a positive growth factor in normal cells, the cells divide. When there is a negative growth factor in normal cells, there is no division. And in cancer cells, whether there is a positive or negative growth factor, the cells divide.